Okay, hi everybody. I've actually never done one of these webinars before, so uh, this is fun. But bear with me if I don't seem like I know what I'm doing at times. So you can see my slides. Yes.
Yes.
So some background. Let's see. I'm Canadian actually, born in Niagara Falls, Ontario, and I went to undergraduate at University of Waterloo in mathematics. Um, Actually, I was in math as an undergraduate, and I love math, but it got to be a little too sort of theoretical for my like. I found it was too abstract at some point, and so then as an undergraduate, I moved into computer science, and started to sort of double major in computer science, which I also enjoy. This was, of course, this is in the '70s when computers were just starting to. We didn't have laptops or desktops really. We had mainframes, but computing was very exciting and at the time. But then after a while, also I found it was too far away from science. So I love math, I love computers, but then statistics I discovered in my fourth year as a student at Waterloo, because some of my friends are taking these courses in statistics, and it was a nice kind of marriage between math and computation, but also it connects to science. So you're using both math and computation, but in the service of science. So that was really nice. And I have to admit, when I first took statistics classes, they were very strange because in math, at least most of the math you start off taking, everything is deterministic and concrete, right? You have rules, and you don't have things that are random. And in statistics, what was unusual was that we talked about random variables and PDFs and things like this, which was very, honestly, for the first year, I thought this was very odd. This is a strange kind of mathematics, but eventually you sort of start to appreciate that way of thinking. And so I fell in love with statistics. Did a master's in Toronto in statistics, and then a PhD here at Stanford with Brad Efron, who's very famous, the inventor of the bootstrap, along with lots of other methods. Went back to Toronto as a professor for 12 years, and then returned as a professor here to Stanford in 1998. So I've been here for almost 20 years as a professor here at Stanford. And I'm in the statistics department where people like Brad Efron is, Trevor Hasty, my long-term collaborator, Jerry Friedman, Percy Diaconis, David Donahoe, a lot of famous people in this department, very good department. But I'm also a member of Biomedical Data Sciences, which is a new department here at Stanford, which has people who do applied work in the biosciences, quantitative work in the biosciences. That department's only a year old, but I'm actually next slide here. I've got this slide here has our web page, and you can see what the formal goal, at least for the department, is to marry rigorous methods development, large-scale biological data sets, and clinical outcomes to uncover the mechanisms of disease and improve health and well-being. So it's a pretty grandiose objective, but it's basically saying we want to use quantitative methods, statistics, and other methods to try to improve public health through data. That's so as at Stanford and a lot of places, there's a lot of excitement in data science for I turn this off, sorry. Well, that works. So,
can I ask a question real quick?
Yes, of course.
What departments are involved in, or who staffs this new biomedical data science department? Is it some statisticians, some clinicians, that type of thing?
Right. So it's a department. It's staffed by biostatisticians at Stanford, and there's biostatisticians are all cross-appointed with statistics, and there's about 6 or 8 of us. And then there are people who do computational biology, genetics. They're all quantitative people. Some from existing departments, radiology, other departments like that. All of who use quantitative methods for health. And these people are sort of in the department in various portions, half, a quarter, etc.
Yeah. Cool.
So this is, I mean, enjoy working at Stanford because, we have a wonderful department of statistics and wonderful students here, but also we have people around the campus who have really interesting problems, and they also appreciate what statisticians can do for them. Right? I find, like, in my collaboration, it's kind of sort of it's almost bimodal in the sense that there's sort of two kinds of people. There's a kind of scientists that meet you and they want a t-test or a sample size from you, a sample size estimate, power calculation by tomorrow, right? Because they're writing a grant or they're resubmitting a grant. And they throw think of the statistician as someone that they need to have on board because the grant says so, right? And that's not the kind you want to spend a lot of time with. I mean, I understand that the need, but it's not very satisfying for the statistician. The other kind of person is a person that actually appreciates what the statistician as a scientist can contribute to the team. And there are people that understand that their data is complex and that statistical methods can really help them to uncover what's important in the data. And they think of you as a collaborator, and I have a number of such collaborators. But when you find a collaborator like that, I find you really have to work hard to keep that collaborator, to work with them, because they're really, it's really special. But it's not just something you do casually. You have to take the time to understand the problems of the collaborators, come up with a solution, not only come up with a solution, but figure out a way to communicate it, right? It's not much use to come up with some complicated mathematical formula and tell the collaborator, I've solved your problem. And then they say, well, can you explain it to me? No, it's really hard equations, but I can write it out for you. Right? That's not going to fly. But it's not just a matter of making the collaborator happy. I find when you're forced to communicate complicated ideas to a non-specialist, you're really forced to understand what's the important parts of this idea. Like, when you strip away the mathematics, what are the important assumptions? What's it really getting at? And so it's a good exercise, not just for the collaborator, but for yourself when you have to explain something in non-technical terms. So for all those reasons, it's a lot of fun being a collaborator, and in the med school here. And I'd spend a lot of my time doing that. So again, you guys should stop me with questions because I don't quite know what I'm doing here. I'm just gonna.
Yeah, this is great. Any questions so far? So we got. He's a part of I guess one of the top, if not the top, statistics departments in the country. It's a very well-known department. Lots of very well-known people there, and he's telling us about this new department at Stanford that brings together multidisciplinary folks whose common denominator is quantitative work in a biomedical setting. So you're hearing things that should sound similar to things you've heard earlier in the semester from our other speakers, like Professor Raymond Carroll talked about, and Jeff Morris talked about the importance of collaboration and how not every collaboration is of the same type or same value from your perspective.
Right.
He mentioned you might have some people who show up when you become professional statisticians who show up and say, I wrote a grant, and according to the grant, I have to provide calculations that show I'm going to have a sufficient sample size to have power to detect something. And that might be what your intended role is. That's it. That's not a very fulfilling or impactful role for you to play. And so what we're hearing from some of these leading statisticians who do this a lot is you want to be looking for the types of collaborations where the person across the table from you views you not as a calculator, but as a collaborator. Jeff Morris said a lot about that. We're not calculators. We're not technicians. We are people who can contribute really valuable stuff to the design of your experiments and how to go about analyzing and interpreting the data. Okay. Any questions so far? I'm guessing it'll be relatively quiet today, Professor Tibshirani, just because there might be some intimidation factor of talking to you. But I'll try to get some questions out of them as we go. So please proceed.
On that topic, I tell my classes, I teach a class in data science and class. I tell them one of the most important skills you can gain as a student is to ask questions. and I have trouble in my own classes getting people to ask questions because people think like the question's a dumb question,
Right.
especially when it's a technical topic. But there are no dumb questions, and if you think you have a dumb question, probably 10 other people have the same question, but they're afraid to ask it, right?
Yeah.
And for me, this hit home when I was first came here as a graduate student to Stanford, and Brad Efron was I hadn't actually met Brad Efron. I'd heard of him. He's very, very famous. And I went to the seminar, and I think 30 seconds in, this guy in the front row saying, what is X, right? And I thought, who is that guy, right? Asking the question. And he said, that's Brad Efron. People told me. I thought, well, if Brad Efron can ask like the most simple, dumb-looking question, right, then everyone else should be able to do that too, and should. So really, it's important to ask questions because then you get, it helps you to get engaged in the topic. Yeah. And a lecture should not be the professor talking to you. It should be a two-way conversation. So that was just an encouragement to get you to ask questions.
All right, thank you.
Oh, no, what else? Okay, so these slides, this is basically a talk, it's a talk about data science that I've given a number of places, and it's got some background slides about data science, and I'm just flipping through quickly the first part of it, just how, in this big data era, what's really changed is the form of the data we get and the size of it, right? In the old days, data used to come as a text file, right? Each row being an observation, a patient, or whatever observing, and the columns being features or predictors. So it's a nice, neat rectangle. It's typically small, right? People would measure maybe 10 things on a bunch of patients. And those 10 things were chosen from their experience about that disease. Nowadays, we collect many more features or predictors than we imagine are even important, just because it's easy to collect things, easy to collect data. It's sitting there often on the web or in a database, and the data doesn't come in a nice, neat rectangle. It might come from the web, from smartphone, from Facebook, a social network, the Twitter feed, doctor's notes, which is sort of free form, right? So just the form of the data is complicated and not simple a rectangle anymore. So that's one thing that's changed. We have a lot more data, way more than we used to have, obviously, and the computation is faster. And how statisticians and computer scientists have responded is to develop new strategies for trying to mine through this data, to model it, to figure out where the important patterns are. So I'm going to talk about one. I was planning on, time permitting, I have a couple of examples of modern statistics in my own work, which I thought I could use as examples. And then the slides here, and then we can see how that goes. And that'll hopefully give you an idea of what the kind of things I'm talking about, kind of projects that I work on, and also try to excite you about the topic. It really is it's a very exciting time to be a statistician or data scientist. It seems like this is just our time now, because there's a convergence of data, computation, and scientific interest in those topics that is all coming together to make the data scientist a key person moving forward. Yeah. There's huge demand for data scientists, as I'm sure you heard from other speakers. In the old days, our graduate students used to go pretty much 2/3 to academia, I'd say. And then some would go to maybe drug companies. Now, probably a third go into academia. When they graduate, become professors. But now another third will go to some of these companies like Facebook, Twitter, Airbnb, Google, of course. Big and small companies. Everybody has data. Everybody's trying to figure out ways to use that data to their advantage. So there's huge opportunities for data scientists in companies, in academia again still, and also in pharma companies where, again, one can collect large amounts of medical data, clinical data on patients, and try to use that to improve their treatments. So I'm sure you've heard same words from other people, but I'm just reinforcing it. It's a wonderful career choice right now, and I don't think it doesn't have a short shelf life, right? Data's going to be around for a long time. Mm-hmm. Science is always going to be important. So I think it's the fact you're in this class is a good sign that you're onto something good here.
Great.
Okay. I'm going to move to an example. Whoops. I was looking to see. I have a little animation here which is not working, so I'll put it on my website though, and I'll give Alan a pointer to it later. Okay. But here's I'm On this slide here, I have some examples, and maybe I'll talk about the first two of them, which are current collaborations of mine. And through those, I'll give an example of some applications of statistics, but also some new tools in statistics which I've helped develop. Okay, so this first example, which is an ongoing collaboration I have with Livia, who was a postdoc here in chemistry, but now she's a professor at University of Texas at Austin, actually. And I visited Livia in her lab a few weeks ago. She's a chemist. Richard Zare is quite a famous chemist here at Stanford. George Pousades is a stomach cancer surgeon, myself, and then some pathologists. So this is, as often as the case, this involves a whole team of people.
Yeah.
And so here's my collaborators, their lab. Okay, so this is basically the description of the problem. So this is about stomach cancer. You guys can see the slides, right? No?
Yes.
So this is like a cartoon drawing of a stomach, and in stomach cancer is treated by surgery, and it's a very bad disease. What the surgeon tries to do is tries to go in, remove the cancerous part of the stomach, and leave as much healthy tissue as possible, so that the patient can have as much functional stomach as possible when the operation's over. And so this is sort of a cartoon drawing. I've got sort of a drawing of a stomach here. The red is cancer, and there's two kinds of normal tissue, stromal and epithelial. Here they are, blue and green. And the idea is the surgeon wants to go in and remove the piece on the right and tries to get all the cancer. So in this case, the edge of the margin of the removed piece is green. So this surgery was successful in the sense that we have all the cancer out. But if this edge or margin was red, then that suggests there might be some cancer left on the edge here. So what happens in the actual operation now is the surgeon removes the piece of this here, his best guess of how much to remove. The patient's still in the operating room. It's given to a pathologist, the piece of tissue. The pathologist in his lab next door examines the piece with his various techniques and determines either, yes, the edge is clear, it's got no cancer on it, in which case he tells the surgeon, you're all done, you can close up. Or if he detects that, in his view, there's cancer on the edge, he'll say, you have to go in and get some more because I'm worried there's still some cancer on this edge. Mm-hmm. So, and this is a difficult thing to do because it's done in real time. The patient's still in the operating room, and the error rate's something like 15 to 20% of this call. And it's also because the pathologist has to do it quickly without his full battery of tests. So the goal of this project is to try to assist the pathologist in making this call more accurately using statistics, machine learning. Okay.
So this is done while the patient is opened up.
Yes.
Yes.
So, okay. So here's what we need is some technology. So this is technology that Livia, my collaborator, brought from Purdue to Stanford, and now is used at Stanford and many other places. It's a kind of mass spec. And frankly, I don't know much. My physics isn't great, so I'll give you a very simple explanation why I understand. But essentially, you put the tissue specimen on the table here, on the slide. An ionized spray is fired at the tissue sample, and that causes free ions to be released from the sample, and they're put into a tube. Okay, this is mass spec, and this is what happens with mass spec is you have a charge. There's a charge on these samples from the released ions, and there's a magnet at the end here. So you fire them down the tube, and you measure the mass. You measure how long it takes for the particles to get from the opening to the end of the detector. And that gives you what's an idea of the mass over charge. So just to give you some idea, so just going back here, so think of each pixel on this tissue. We get a measurement, which is one of these spectra. So each pixel is giving us, see this little pixel here, is giving us a spectrum. What are these things? This is mass over charge, but think of it basically like you've got a little soup with various amounts of ions of different types. And this is telling you that the abundance of each of the ingredients. So that, for example, there's 100 at 281.33. So for the chemist, that's telling you we have a lot of this ion. We have a lot less of this ion. But it basically, it's a way to think of it to tell you the relative abundance of ions in the pixel. Okay. So this is for a cancerous pixel. This pixel is known to be cancer. Okay. This is another pixel that's normal. Okay. It's this pixel here, and you can see the profile looks different. How does it look different? Well, it looks like maybe it's a little lower here, a little lower here, a little higher here, right? Mm-hmm. And here's another normal, a stromal pixel. It's different again, right? So the goal is to figure out, can we use this profile of peak heights to distinguish a cancerous pixel from normal pixels?
Let me make a comment for the students' benefit real quick here. We have, this is multivariate data, so you don't know a lot about that yet unless you're taking some upper level classes, but for each, quote, individual in the data set, you don't just have a single response variable that you're seeing. Like
you have a single response variable, which is the cancer response. Yeah, but you don't have a single feature vector, right? You have a feature of an entire vector of features.
Right. Yeah.
Right.
So for each individual, we have this whole spectrum or spectra that he's showing for each one of them.
And for each pixel, for each individual, like this is Mr. Smith's cancer sample. For each pixel, we're gonna have one of these, but we're gonna have like a few hundred pixels measured for this guy, in all different places.
So it's actually a very high dimensional for each. Individual, there's multiple spectra for the sample of tissue. Okay.
Right.
All right.
Okay. So imagine, before we get into the stats, like how we model this. Imagine. So this is what our data looks like. We actually have, in this initial study, we have 20 patients, each one contributing a piece of cancer tissue in each of the normal tissues. And then for each of these tissues, there's pixels and all these measurements. Okay. So then the question, somehow we want to develop a rule that uses the pattern of peaks here, the peak heights, to distinguish cancer from normal. That's our goal, right? So that a new patient sample comes in, he's in the operating room, we get his sample, we find these spectra, using the machine. We put the sample on the machine in real time. We get the spectra, and now we have to develop a rule that says, I think the machine has to say, I think that's cancer, or I think that's normal. So is the goal clear?
What do you think? Everything okay? Yeah, lots of nodding heads. It's predictive modeling. It's statistical learning, like we've talked about in our classes.
Before we get any kind of fancy model, we can think about how do we do this just if we didn't know any statistics? How do we do suppose we just, we were pretty good at computing. We could collect all the data. We had we have, let's say, a few hundred of these kinds of profiles for cancer and a few hundred of these, and a new pixel comes in from the new patient, and you're asked to determine, is it cancer or not? What would you do? I'm asking you guys, what would you do?
Okay, so let's think about that. Suppose they have this data set for each of a bunch of individuals. They have tissue samples and multiple spectra for each of those tissue samples. Right. And so an expert has visually inspected each one of the samples and told you this is cancer, this is not cancer.
Right.
So you have what we've called training data. Right. So suppose a new patient is rolled into the operating room and they extract a tissue sample and they give you a mass spectra profile. Dr. Tibshirani is asking, just based on intuition, what would you do? How would you go about making the decision, is this a cancerous one or a non-cancerous one or what? Kurt, go ahead.
Compare the new one to the data you have and see which one it matches or looks the closest to.
Yeah, which one it looks closest to is a reasonable idea. Emma, go ahead. Yeah, go ahead.
Sorry, no, go ahead. No.
I was asking you to go ahead. Yeah, please.
As in me. Okay. So
Yeah.
I was going to just, as a follow-up, you said closest to it. How would you measure close?
Yeah, so how would you measure close? We have peak heights for a bunch of locations on the x-axis of this spectra. I have peak heights for this new tissue sample that I'm trying to predict, and I have peak heights maybe at the same locations for my training data. How would you define close, do you think? Not just Kirk. Who would like to answer? Go ahead, Emma.
Would you do a hypothesis test to see if it was like statistically significant, the difference between the peaks that couldn't be explained by natural variation?
Okay, so we could think about doing a hypothesis test, asking, are the differences between this new individual's peak heights statistically significantly different from the cancer one? Okay. What about just asking distance? What's the distance, or how close is, say, my spectra to that cancer spectra? What do you think? Clustering algorithm? That's a good idea.
Yeah, I mean,
Go ahead,
well,
Professor Tibshirani, fill us in.
yeah, no, I mean, clustering is, you've almost got it. I mean, when you do clustering, you have to assign how far objects are from each other. And the usual metric is the square distance, right? So, like, for each of these pixels, I have a vector of peak heights. I suppose there is about 10,000 of these, right? This is going to be sampled along a very fine grid. I have a vector of 10,000 peak heights, and for each of these pixels. And now my new sample comes in. I can measure its distance by saying how far are the 10,000 is the vector in 10,000 dimensional space from each of these guys. I can just find the square distance between those 10,000 numbers, the new ones, and 10,000 of each of the old ones, right? And just take the sum of the squares over the 10,000.
So like a vector distance.
A vector distance.
Yeah.
Okay, so that's pretty good. And actually, that is what's called nearest neighbor classification. You've taken that, or if not, that has an official name. It's nearest neighbors. You predict classes by finding the nearest neighbors in the existing classes, and you decide to classify by whichever it's closest to. So that's the idea that you guys already said. So we just put a little bit of meat on that idea. But what's lacking there is that, we suspect that most of the peaks aren't going to be important, right? I mean, if we were really fortunate, then there'd be one sort of holy grail peak, right? Like maybe this one or something. Like that all that mattered was the peak height at, say, 281.3. If you're high on that, you're cancer, and if you're low, you're normal. That's possible, right? It looks like that, that may be the case here. Well, at least from these 3 pixels. But more likely, there's probably some panel of peak heights, some group of them that are important, but we don't think all 10,000 of them are likely to be important, right? We hope not. So I'm going to show you a method, the lasso, which is something I developed a number of years ago and is used a lot, which gives you what's called a sparse model. So it's going to try to pick and choose the most important peak heights, and we're going to end up with, starting with 10,000 peaks, we're going to end up with just about 100 of them. And those 100 are the ones that are going to tell us the most about the contrast between cancer and normal. That's called a sparse model. So that's a pretty big area of research now, sparsity. Try to take a large set of features like our 10,000 peak heights and pare them down to a smaller number that have the signal that we're looking for. Okay? And there's 2 reasons I want to do that. First is a matter of separating the signal from the noise. Like in that pattern matching we just described using all 10,000 features, if most of the features are just noisy, that they're not actually contributing, they're going to add in noise to that metric, right? And that's going to confuse the classifier. Second of all, not only do we want to classify here to help the patients, but the scientist wants to learn about which of these peaks are important because they want to understand the process of stomach cancer, right? To the chemist, this actually means something, this peak. He can do another test and figure out just what ion is at this site. So this means something to him from a chemical point of view, and that can help to understand the process of cancer and maybe eventually hope to treat the cancer, to find new ways to treat it. So the goal here is two-pronged. One is to do good predictions, and the other is to try to come up with a simple model, a simple set of biomarkers, a subset of these, which can help us to understand the disease. Okay, so.
Everybody okay? Any questions?
Again, please ask questions if there are, because.
Okay, I think we're good. I think we're good right now.
Okay, so this is a little more advanced now, but I'll just try to give you an intuitive idea of what's going on. Okay? So the lasso is a method that, again, I developed a number of years ago, and it's used quite widely now, and it turned out to be a really good tool for this particular problem. So you guys have taken least squares regression, is that right, Al?
Yes.
Okay, so everybody's done regression. So just remind you what regression is. Regression is when you're trying to predict a response variable from a bunch of features, predictors, right? And so you postulate a linear model between the outcome and the features. So here I've written, for the moment I'm ignoring this last part. I'm just reviewing, this first part is least squares regression. I have a set of features. I'm going to find a set of weights, beta j, so that the predictions, beta 0, well, plus, because I have a minus, the discrepancy between the actual outcome and the predicted outcome is as small as possible. That's least squares regression, right? So given a set of training data, features, and the I indexes the observations and j indexes the feature. Given the features and given the outcomes, least squares says, find me the weights that make this discrepancy as small as possible. It's just, and I was trying to find a set of weights that allow my predictions to match the outcome as closely as possible. That's least squares regression. So that, in one dimension, that's fitting a line. In higher dimensions, you're fitting a plane, a hyperplane to data, right? That's least squares regression. Probably it is the most used model in all of statistics. Okay, it's been around since Gauss, I think, way back, whatever that, 1700s. Mm-hmm. Now, for the moment, I'm moving away from our problem. In our problem, the outcomes are actually classes, right? There's cancer and normal. But for the moment, I'm just making it simpler. I'm thinking of a continuous quantitative response like age or blood pressure, something the yi is not 0, 1, 2, but it's actually a continuous variable. And that's a simpler application of regression. Okay, so this is least squares regression. But least squares regression is a wonderful method, but suppose, here's a place where it breaks down. Let's suppose I've got more features than there are observations. In other words, I've got 100 observations, that's the index I have 1,000 features. Right? Now, if I tell you, find the set of weights to match so that the predictions match the outcome, you're going to be able to fit it perfectly. You're going to overfit. For those of you who have taken linear algebra, that's a singular system of equations, right? There's no unique solution because you have more parameters than you have observations. You can perfectly match the predictions to the outcome, which might sound good, but it's bad because that's what's called overfitting. Right, so the weights you'd find would look really good on your data. They'll give you 0 error. But if you tried it on new data to predict the outcomes for new test data, a separate validation set, you'd find it predicted very badly because it's overfit to the data. It's responding to all the noise in your particular data set. It's not picking out the signal. It's also matching noise. So that's overfitting. Yeah. I don't know if you guys. I can pause for a second. That's a really important concept.
We have seen some of that. Some of us in here have seen that in another class. Some of us probably have not. So this is good for them to hear maybe for the first time.
And this is going to occur in our problem, our cancer problem, because we have actually, I think the number of pixels in our data sets like 6,000. Number of features, which are peaks, is like 10,000. So there's more peaks than there are pixels. So if we just did if we did this kind of matching, we would overfit. Okay. Yeah. So we need to do something to basically constrain the regression to fit, so as to avoid overfitting. And the lasso is a popular method for doing that. Here's the lasso. It's least squares, but there's an extra piece. Subject to summation beta j less than s. This is like a budget. So think of it this way. We have our data, x and y, and all these observations. And at first I say, okay, could you please do for me least squares? Go ahead. Okay, so you run least squares, and you say, Dr. Tibshirani, I'm really happy I got an error of 0. I say, hey, wait a minute, you've overfit. That's no good. That's not useful to me. So I'm going to change the rules of the game. I'm going to give you a budget now. And so you can't pick any old betas you want. I'm going to put a budget on the sum of the absolute values of those coefficients. And now I'll say, class, please go do the fit, but you have to do it under budget. So for example, if the S was 1, and I'll tell you in a moment how we pick a good budget, but if the S were 1, your job is to find the best fit as possible with a total budget of absolute value of beta j's as at most 1. So what is that going to do? Let's see. Actually, let me ask you, how is that going to affect your choice of betas?
Yeah, so suppose you fit a linear regression model, or in this case, maybe a logistic regression model or something like that.
I mean, we'll stick to this particular model, but we're going to circle back to logistic. But
Okay, so let's do linear regression. You have a numeric response,
yeah.
and you have a whole bunch of predictor variables. For each of those predictor variables, you're going to allow there to be a beta coefficient in the model. So you're saying my response, on average, is beta 0 plus beta 1 times predictor 1 plus beta 2 times predictor 2 plus ... all the way through all your predictor variables. Multiple regression.
Right.
If you don't impose this penalty or budget that he's talking about, then you would do usual least squares. Find among all the beta coefficients the ones that correspond to the best fitting line, least squares style. What if we said, okay, do that, but at the same time, you have to have the sum of the absolute values of the betas no bigger than a particular chosen number.
Right.
What is that gonna do? Suppose you hit enter in R and ask it to do that. What do you think is going to be the difference between the model with this budget and the model without it?
And I'm asking sort of in a qualitative way. I'm not asking you to actually, tell me exactly how you numerically do that, but from an intuitive point of view, qualitatively, what's this going to do?
Yeah. What do you think? Think about the beta coefficients. We have two models. One is without the budget. One is with the budget. What's going to be different about the beta coefficients generally in the model with the budget, do you think?
The ones that you chose to minimize will be smaller?
Yeah, it'll be smaller in the sense that they're closer to 0, right? Because we're putting a budget on the sum of the absolute values. Yeah.
Right.
Okay. So the coefficients are going to tend to be kind of shrunken towards 0.
And
Make
I mean,
sense? Go ahead.
right. And which ones, if you had to guess which ones were, so some will be non-zero and some actually turns out are exactly 0. But can you which kind of predictors would end up with the non-zero weights?
Yeah, so suppose you do this.
Yeah.
As he said earlier, if we had thousands of features, each of these locations on the x-axis of the mass spectra, for example, surely a subset of those are not useful for predicting. There's almost certainly a subset of them that are just like noisy variables. They're not going to help.
Right.
Some other ones are going to be informative. When it's a big peak at this location, that tends to be cancer. When it's a low peak, it tends to not be cancer.
Right.
So what type of those predictor variables are going to wind up with big beta coefficients and absolute value, do you think? What if we had a location, a predictor variable that was noise? It was completely irrelevant, did not help us do prediction at all. What would you hope its beta coefficient would look like after doing a budget kind of thing like this? 0, right? You would hope that it went away.
Yeah.
So which of the variables do you think are going to have the big betas in absolute value? The significant ones, yeah, the ones that are informative, right?
Right. Like for example, maybe here, again, I guess maybe the peak height at 281.33 looks like it's quite different between cancer and normal. So, right. So, I mean, you can think of this basically as, in this optimization with the budget, you're going to, if you were the optimizer and I gave you a budget, you're going to spend the parameter. You have a certain budget, you're going to spend them where you get the most bang for the buck, right? And that bang for the buck comes from, you're going to put non-zero weights on features that actually uh, are different between well, in this case, it's a continuous response, but those which are correlated with y and predict y, those are where you're going to put the non-zero weights. The ones that are just noise and have no contribution, no relationship to y, you're not going to spend your money there. You're going to give them zero weight, right? So that's basically the idea. We give it a budget, and it's forced to put the non-zero weights where they matter most, on the features that predict y the best.
Yeah.
That makes sense?
Okay, we've got a question here. Emma, go ahead.
So you're basically trying to quantify the Blotto principle that 20% of your inputs will account for 80% of your output. Like you're trying to find the ones that are most important to predict like your the differences between the two types of cells.
Yeah, so one of our students is citing the 20-80 rule, I guess, where 20% of, in this case, features perhaps are the ones that are interesting, and 80% maybe are the uninteresting ones, and we're trying to do that. I think that's a good analogy. What do you think, Professor Tibshirani?
No, I've never heard that particular rule. It could be 20-80, but it could also be, right, it could be 5-80, or right. I mean, that in general, that's a good analogy. You don't need to take the 20-80 exactly,
right,
literally, because what we're going to do, or what we do is let me step back. So for a given budget, like S equals 1, for example, we have to minimize this squared error under this budget. That turns out to be what's called a convex numerical problem. So that's a very well solved computational problem. And in R, we have a package called GlimNet. There's a lot of other software around. Basically, you can give it the data, give it a budget, and it will give you the betas, the weights that best minimize the sum of squares under the budget. So that's one important thing is we can solve this problem. Like on my Mac laptop, for example, I can solve the problem for this cancer application in about a minute using GlimNet. So it's quite remarkable. Although there's like 10,000 features and 1,000 observations, and for a given budget, you can solve this problem very quickly on a standard computer. So you can, after class, you can download GlimNet, and you can try this from CRAN, our language repository, and you can solve this very quickly using this program. So then the question is, maybe a remaining question: how do we pick the budget? As the one student said, 20-80, for example, like should the budget restrict the proportion of betas to non-zero to be 20%? That was a suggestion of the 20-80 rule, right?
Yeah.
Well, we don't know for sure. Maybe it's 20-80, maybe it's 10, or maybe it's 50. We don't actually know for sure. So what we use was called cross-validation. So just in words, what we do, this program does, you divide the data up into like 10 parts. I mean, the observations into 10 parts at random. You basically fit this lasso model for various budgets on the 9/10, that's the training set. And you take that model and you predict the data you left out. And do that for different budgets. And you see which level of budget gives me the best predictions on the left out data. Okay, and that's really important because that's the budget we want to use. We want to use the budget that for new data leads our model to predict the best. Right? And there's no way ahead of time to know what the right budget is, because, for example, if you have very noisy data and there's only a very small number of important features, you're going to want a tight budget, right? You can want a budget that maybe only gives you 5 non-zero weights. On the other hand, if there's a huge amount of signal in your data, many peaks that are informative, maybe hundreds of them, you're going to want a bigger budget. Right? But we can't know that ahead of time. That information is in the data. But cross-validation gives us a way, based on the data, to pick the budget. And again, you try a range of budgets, and you choose the one that leads to best predictions on the data you left out of your sample. So that was a very quick description of cross-validation, but it's really important for all of these statistical learning methods. They all have some kind of what's called a tuning parameter, which adjusts how hard they fit to the data. In this case, it's our budget. But any other model you run into, like deep learning or support vector machines, or there's a whole battery of new methods out there. They all have tuning parameters which control how complex they can a model they can use.
If for you students, if you're in the 489 class that I'm teaching on data science right now, we've talked about linear discriminant analysis and logistic regression as classification methods.
Yeah.
And we've, quote, tuned those by considering what probability threshold above which should we predict yes. If the estimated probability that a baseball player is in the Hall of Fame is bigger than 0.5, should I predict Hall of Fame? Or should I make the threshold smaller or bigger? That's the same idea of quote, tuning a predictive model.
Right. So again, getting back to the student's question, it's really asking, should we use a 20% rule or a 10% rule or a 50% rule for this data? Yeah. Let me just circle back now to our data. So this gets a little complicated, but our data, the outcome is not continuous like the Y on this slide here, right? I mentioned maybe blood pressure, age, or something like that, life survival time. Remember, our outcome is one of these three classes: cancer, or the two normal classes. So we use what's called a Loggia kind of model, logistic model, which models the probability of being one of these three classes, given the feature vector. Feature vector, again, is the height of the spectrum, these peak heights. So again, I don't need to go through this as a detail, but just at a high level, what we're doing, we're going to take our data, we're going to model the probabilities with a bunch of weights, and we're going to subject these weights to this lasso penalty again, with a budget. Okay, and we're going to pick the budget using cross-validation to best avoid overfitting, just to get the sort of a sweet spot typically between fitting and overfitting. Right? If the budget's too tight, the model's too constrained, it can't do anything. If the budget's too big, the model can overfit. There's usually a nice sweet spot in the middle where you just do just the right amount of fitting. You pick the features out that have important information about your data, and you set the rest to zero. That's kind of the sweet spot you're looking for in the budget. Okay, so I'm not expecting you to understand all these details if you haven't seen them before, but at a high level, we're going to fit to our data with weights. And by the way, what are these weights? Beta, J, K. J is the feature, and there's 10,000 of them. Those peak heights, and K is 1, 2, or 3. Epithelial, stromal, cancer. So we get for each class, for each feature, we have a weight. And the overall sum of the absolute values of the weights are subject to a budget, which we're going to estimate by cross-validation. So at a high level, it's really the same as what happened here, except our objective function is we don't have a continuous response, we have a class-valued, a categorical response, right? Cancer, epithelial, stromal. Okay, so it's great because now we basically can take the data and put it into this program called GlimNet, which fits the lasso to this data. Okay? And finally, some results. So here's what happened. Okay, so there's actually two sets of results because there's two, well, I had 20 patients here in this initial sample. Each patient is contributing a bunch of pixels, a few hundred pixels from each of the kinds of tissue. And by the important fact I forgot to mention, but no one asked this. I said that the classification into normal or cancer by the pathologist was quite difficult, right? The error rate was like 15 to 20% at the time of the operation. But what's happened here is we've taken the samples, and this is post hoc. This is after the operation's over. The pathologist has spent a couple weeks in the lab doing various stains and tests on the sample and getting a much better idea of whether it's cancer or normal. So these labels, these true labels, are actually pretty much a gold standard. I mean, they could be wrong, but they're almost surely right because the pathologist had 2 weeks of tests that he or she has applied to the sample. Okay. So as opposed to the labels at the time of the operation, which are challenging to get, right? This is a long, okay. So here is results of cross-validation. So remember, we take our model, we fit it for various budgets, and then we look to see how well we predict the left-out data. and we pick the budget that produces the best prediction. So here's the result of the cross-validation. The rows are the true labels. So each of these are pixels, right? These are pixels which were decided by the pathologist are epithelial, cancer, and stromal. The rows are the true labels. The columns are what the lasso algorithm predicts. Okay? And this is called a confusion matrix. Okay? That's for obvious reasons. It gets about 89% correct. Pretty good. And in each of the classes, here's what it gets: 95, 83, 96. So it's close to 90% correct. And if we actually collapse to see stromal, if we just look at cancer versus normal, like if you think of this as a 2-class problem, if you do the math here, you get an error rate about 95% correct for the 2-class problem, cancer versus either of the other two. Okay. But we also kept 6 patients aside as a separate test set because we were worried, well, we need lots of models here, we might overfit, and let's have a separate validation to see how well does the model fit on work on absolutely new data. So here's 6 new patients. Same model we take here, we just apply it to this new set of 6 patients. It's getting about 96% correct. So very encouraging. These are really good results. We're getting 90 to 95% correct on 2 separate validations. And here's the sparsity. The budget it shows led to only 129 peaks that were non-zero. Remember, it chose the best budget. It decided that you only need 129 peaks to make this prediction. All the rest, 10,000 or so, are thrown away. They're all zero. So we pared down a complicated problem with 10,000 potential peaks to only 129 of them. And that was 129 are doing a good job of prediction. So our predictions are quite good.
Is that clear? Everybody okay with that so far? Kind of done a feature selection step in a way. Yeah.
Right. This budget has forced a lot of these guys to be zero. It chose a pretty tight budget because it felt that was the most advantageous. And the result is that, the grand majority of peaks weights were set to zero.
Great.
And it put its money where it tried to find the most bang for the buck. And this is where it decided to spend its money on these 129 peaks. And, I don't have them here, but there's a list of those peaks. And to the chemist, they mean something, right? So they can look to see, this is saying that peak number 48 is higher in cancer. Let's look to see what's at that peak, what chemicals there. And they can then try to use that to understand how cancer and normal are different for this disease.
Mm-hmm. Cool.
And so we've taken a complicated data set and we've come up with quite a good prediction rule. And what it's doing is sort of what I asked you the students, how would you do this? You guys said pattern matching. Well, this is basically an advanced form of pattern matching, right? Except it's not using all the peaks. It's determining, which 129 are the most informative, and then it's doing kind of pattern matching among those 129. So it's really, it agrees with our intuition, but it's just doing a little more for us.
Mm-hmm.
What else do I want to say? Okay. I'm going to skip to. This is, I think, interesting. So this is an example to me of why I'd love to work in applied statistics with collaborations, because it's hard as a statistician to sit at your desk and figure out what's an important research topic, right? And actually, I think statisticians in the 60s, Brad Efron, who was my supervisor, he confessed this to me. He said, when I was a young professor, I used to sort of sit at my desk and try to figure out what should I work on, right? This is before he sort of talked to scientists or collaborate. And now 10 or 20 years later, he said, I have no idea how I could do that, right? The point being that it's only by talking to scientists that you really understand what are important, what important problems to work on.
Mm-hmm.
And here's an example, actually, in this very project. This is a problem I haven't solved yet, and I'm working with graduate students on this, but It's difficult, but clearly important. So, we built this classifier. It works. You saw the results. Looks quite good. We're now testing on many more diseases, actually pancreatic cancer and prostate cancer, skin cancer. It's doing quite well in general. But in stomach cancer, my collaborator, after we had trained the models and we had the results and we were writing up the paper, she came to me with another sample from a patient. Again, not the actual physical sample. She brought me the spectra, right? And she said, Rob, could you put it through your classifier? Well, please tell me what your classifier predicts for this sample. So I did that. I just applied the weights, and it said, the classifier said, I'm quite sure it's cancer. Remember, I didn't say this, but this classifier gives you not only a prediction, but it tells you a probability. So it not only says it's cancer or not, but it says with what probability do you think it's cancer? And that's important, right? If a classifier says, well, I think it's cancer with probability 0.99, right? That means a lot more to you than if it says, I think it's cancer with probability 0.51, right? The second case, it's really not very sure. In the first case, it's very sure, right? So those probabilities are important too. Well, in this case, she brought me the sample, and it said, the classifier said, I'm really sure it's cancer. It said like 0.85 or something. So she said, oh, that's interesting. And then she brought it to the pathologist, who then did 2 weeks of testing, and the pathologist said, I'm quite sure this is normal. So the two didn't agree. So who do you believe, the man or machine, right? So then Olivia looked more closely at this and at the sample, and it actually had a lot more fatty tissue than any of the training samples I had seen, that she had seen, right? So perhaps this had metastasized from some other tissue or something. So it was very different physically than the training sample. So what happened was, I postulate that the classifier was being asked to extrapolate. Here's what I mean. This is like a little picture of this. Whoops. Imagine, I just want to move this down a bit. Just a bit. Oh, well, I can't seem to do that. Okay, that's okay. So these are two features. Remember, our features are 10,000 dimensional, but for the purpose of describing the problem, I've said, imagine we have two features, just two peak heights. And here's two classes, right? The green, let's say the green is normal and the blue is cancer. Okay. And we have here's a situation where there's a really nice classification, a nice distinction between blue and green. It's just, we could just draw a line like this maybe, right? And things to the northeast are blue and things to the southwest are green. Okay. And now you bring a new sample in, a query point, here the X, and you say, Rob, could you tell me, is it green or is it blue? Well, I don't know about you guys, but you probably say, oh, it looks like it's probably cast green, right? Because it's closer to green than it is to blue. But now suppose I were to move the query point way up here to the northwest. Okay, let's say right there. What would you say now? Well, what the classifier would probably say is the classifier basically does a nearest neighbor, essentially match. So it'd say, well, gee, this point is much closer to the green than it is to the blue. So it would say, I'm quite sure it's class green, right? Because the relative distance from this point to the greens is much less than that than it is to the blues. But do you believe that? I hope you're worried. Like if you saw that, you think, gee, maybe it's right, but you're really extrapolating here because I have no data out here, right? There's no data. So it may be that this decision boundary can be extended way up here, but maybe I have reason to worry that this is actually an extrapolation that's unwarranted, right? So I think that's what happened in this example. This fatty tissue, in a sense, was, as a point in 10,000 dimensional feature space, was far away from the training data, right? Because it's very different, its composition. So you have to imagine this picture in 10,000 dimensional space, okay? Not 2-dimensional, 10,000 dimensional space, right? And we have a point which is far away from all the other points. Of course, all points are far away from every other point in 10,000 dimensional space, but in some sense it's far away, and we've asked the classifier to extrapolate. And that is fraught with danger. And so the point is, if we want to actually put this into clinical practice, which we do eventually, the classifier has to be able to abstain sometimes. So we want to arrange so that the sample might come in, and some of the time the classifier will say, I'm not willing to make a call here. I have to abstain. This is too different from what I've ever seen. Like with the fatty tissue, right? In that case where it probably had no right to make a call. That was a dangerous thing to do. The question is, how do you design a system to do that in an automatic way, in a way that's reliable? First of all, in a way that doesn't happen all the time. If it's doing that most of the time, it's not going to be useful to you, right? But if you want it to abstain occasionally in situations where it should abstain because the point is too far from the training sample. So this is a really interesting problem, and I have no idea how to solve it. And I don't think anyone has solved this problem in high dimensions. But clearly, for this kind of application, it's crucial because you can't have a classifier making calls willy-nilly when it really, in situations where it doesn't have the information.
Yeah.
If you were a patient, you wouldn't want the system to make calls, if it wasn't sure. Basically, right? So here's an example of how a really important problem, which I'm now working on with my PhD students in statistics, was I wouldn't have thought of if I hadn't been involved in this application. But it's clearly a key question for this particular application.
Cool. So just FYI to your students, if you're thinking about graduate school, which I hope you are, this is kind of the genesis of a research problem.
Right.
You work with a faculty member who's an expert, and they're collaborating with people who are doing real-world applications, and they uncover a case that, hey, we don't know what to do here. There's no method yet. So you, as a graduate student, might get tasked with figure this out and deal with it rigorously, and maybe write some software and write some papers.
And the thing that's nice is if you solve the problem, you're not going to just solve it for this particular application, right?
Yeah.
Which is good. Hopefully your solution will be somewhat general. That's the nice thing about statistics, is that it's sort of high up on we call it the tree of science, right? So that if you come up with a method for detecting outlying points in high dimensions, it's going to be useful for many domains, many applications.
Yeah.
So it came from this particular application, but the solution will be useful, hopefully, for many different applications.
Yeah.
That's also why building software is such an important part now of scientific our scientific work, like the GLIMNet program I mentioned in R, which does this analysis of these sparse models, is an R package which we've spent a lot of time writing and supporting, because people use, the reality of it is everybody's busy, right? And if you publish a wonderful paper with lots of different, lots of great ideas, if there's no software for doing that, if there's not good software for carrying out that idea, no one's going to use it, right? It's going to be published in a journal. You're going to be happy for a few hours. You see the paper, and people are going to forget about it, because if you go to a paper and you think that's a good idea, and there's no software, then what's your choice? Your choice is to write your own program, but that's difficult, and we're all too busy. More likely, you're going to look at some other method someone's come up with. If they've got software, you're going to use it, right? Because whatever you're doing in your job, no one has enough time to develop software on their own. So it's really important to develop high quality software and to support it in an ongoing way, because that's how you transfer knowledge into science in a way that people can use it effectively.
Cool. If I could, I'd like to take a few minutes. We've got about 9 minutes until the classroom is no longer ours. So if I could ask you, what kinds of general advice do you have for young statisticians, just generally for career advice? What trends do you see on the horizon? What kinds of preparation do you think would be most valuable? What kinds of career trajectories do you see as interesting and high potential in the upcoming 10, 15 years or so? What do you forecast?
Well, let's see. I guess, I mean, I'm talking from the point of view of a statistician. So, when I talk about data science, statistics is only one sort of part of that hub, but I think it's a really key part. So, the trends are sort of some of which I mentioned in the beginning. The data is getting more complicated. It's getting bigger. It's harder to deal with. Despite that, I think that there's certain principles that in statistics that are never going to become, that'll always be important, like how to design a good experiment, ideas of bias and variance, overfitting and overfitting. All these things are important. So I think for that reason, it's whatever you plan to do in data science, getting a degree, well, an undergraduate, maybe a master's degree in statistics, or a PhD if you have the energy and desire for that. That's always going to serve you well, because the principles you learn, how to approach a problem, again, the kind of principles like bias and variance and randomization and all these things, they're always going to be important. I mean, the way in which they're applied will differ. The scale at which they'll be applied will be different, but the concepts are always going to be important. So I would definitely think that's a good idea to get some good, solid background in traditional statistical concepts. But to add to that now, that's what I did in the 80s when I got my degree. What's different now? You also have to know a lot about computation, which we didn't have to know so much about. So the other part of it is learn a lot about computation, learn about distributed computing, learn how to write a serious program, a software program. Learn whatever the languages are. I mean, R, but also maybe C++ or Java or Julia. Just learn good principles of programming. Learn about distributed computing, parallel computing, because that's becoming important as the data is too large to put on a given machine. So learn about traditional statistical theory, be good at computation, and I think that combination is unstoppable, really. Because there's going to be a huge demand for people that have those skills.
Yeah.
So.
Cool, thank you. Any questions right now from you guys and gals? Asaf, go ahead.
I was wondering, how often are new methods developed just through theory versus application? So like in your case with the cancer data.
So you say, how often are
how do you break down, I guess, the frequency or rate at which new methods come out based on theory alone versus being motivated by some specific real-world application?
That's really hard to tell, right? Because very hard to tell. I think more and more it's the latter.
Yeah.
But there are still good ideas that sort of emerge out of theory.
Mm-hmm.
Yeah.
Okay. Thank you. Any other questions? Taylor, go ahead.
Yeah, going back to the cancer, removing cancer cells,
Yes.
can you talk more about the difference between like making a type 1 error and a type 2 error? Because you want to like make sure that you remove all the cells, right? Because if you even leave a little bit, the cancer could come back. So could you talk about like how to make you were making a linear model earlier, how to account for the probability that there are still some cancer cells left?
Yeah, so if I may kind of summarize, how would you comment about balancing the different error types in this context?
So that's a very good question. I actually, it's a detail that I neglected to say, but what I actually did here was the objective function you minimized wasn't simply the misclassification error rate. Rather, I put twice the weight on the cancer class. So in other words, the error rate was the error rate being minimized by the lasso was the error in the normal class plus twice the errors in the cancer class. In other words, I penalize the algorithm twice as much, twice as much if it made an error in the true cancer class versus made an error in the other classes. Mm-hmm. That kind of answers your question, because and what that did was, by the way, it made this error better and made these a little worse, right? Because I think the point you're making was a good one, is that you can make two kinds of errors. You can classify a truly cancerous pixel to normal, and that's bad. It's really bad because it means that, for example, in this application, the surgeon might be told, okay, you're all done, and in fact leave cancer in the body. And then the other kind of error is when you call truly normal pixels cancer. That's not as bad, right? Because it means the surgeon will go back and maybe take a little more more tissue out of the body, which is not good, but it's not as bad as leaving cancer in the body. So the errors are not symmetric here, and I put a penalty, 2-to-1 penalty on the cancer errors.
Cool.
And that was sort of arbitrary, but I did that, and I looked to see how these relative percent corrects varied as I increased that weight.
We talked in the data science class about balanced accuracy, and I mentioned that you can do a weighted average of sensitivity and specificity, for example, with the 2-class problems that we were playing with. So it's similar things that he's doing.